So thank you very much for letting me visit again, and for the Guy Foundation for sponsoring this great series. It's an area of great interest to me because I work on the mitochondria, and the mitochondria generate about 90% of the energy of our bodies, and they do it in a very sophisticated biophysical manner. And I believe this would be a very central place for us to understand not only the role of physics in biology, but also hopefully or possibly quantum biology. So the hypothesis that I'm going to put forward, pardon me, is that mitochondria can communicate through electromagnetic radiation, and that quantum functions are important in bioenergetics. And I would like to call your attention to the left slide, where you see some mitochondria, and you see the thin lines going into the center of the mitochondria called cristae. And those cristae, as you can see, between different mitochondria are aligned, so that the mitochondrial cristae of one mitochondrion actually seem to flow with the cristae of the other adjacent mitochondria. Now, these mitochondria are linked together by something that looks like a synapse, although we really don't know what glues them together. But as far as we know, there's no physical interaction. There's no direct chemical interaction between the two adjacent mitochondria. So then the question is, why are the cristae aligned? And I'm going to argue that the cristae are aligned because of electrostatic repulsion. I want to mention much of the work that I'm going to mention today have been done with my close friend and collaborator, Peter Burke, at the University of California, Irvine, where we started this work more than 20 plus years ago. So to summarize, if we diagram these cristae, they're infoldings into the mitochondrial intermembrane. There's an outer membrane, an intermembrane space, and an inner membrane. And as you can see, if this is a double membrane, the amount of lumen between those double membranes is less than the width of a membrane, which is less than 0.2 nanometers. So if we then diagram this, we've shown that, in fact, these cristae lumen are closed compartments, and they're closed by a series of proteins called OPA1 and mucose, which I went into in my talk. And the electron transport chain, which pumps protons across the mitochondrial inner membrane, is all arrayed along these lipid bilayers of the cristae. So they pump the protons into this very, very tiny volume, the cristae lumen. When you look at the ATP synthase, which uses this electrochemical gradient to generate ATP, you'll see that the ATP synthases, which are dimers, are always aligned to the outer edges of the cristae, where the maximum electrostatic repulsion is driving the protons through the spinning wheel that generates the ATP and the ATP synthase. Now, what we also have observed is that there's another protein in the mitochondrial inner membrane, in these cristae inner membranes, called the adenine nucleotide translocator. And the adenine nucleotide translocator was famous for 50 years because it exchanges ATP and ADP across the inner membrane, and therefore is the vehicle by which ATP gets out of the mitochondria. If that's the case, which we do know it's true here, what is it doing here? Because there's no point in putting ATP in the cristae lumen. There's nothing there for the ATP to work on. So what we've discovered, as you'll see, is that the adenine nucleotide translocator is bifunctional. When it's out here, it exchanges ATP and ADP. But when it's here, it acts as a voltage-dependent proton channel. And that is basically a rectifier. It keeps the membrane potential at about 180 millivolts. As soon as it gets to that level, it opens the proton channel, and then the protons are released. And if you look at the mitochondria with a voltage-dependent dye, you can see that the cristae are the areas where you have the maximum electrostatic potential. So basically then, the way the mitochondria makes energy is it burns hydrogen, in this case carried on the carrier NADH, to give you NAD, and the electrons move through complex 1, coenzyme Q, to complex 3, to complex 4. And as the electrons move through these systems, they pump protons across the inner membrane into that cristae lumen. And then the protons can either be used by the ATP synthase to make ATP, or, as I said, when the voltage gets too high, the adenine nucleotide translocator releases the protons back into the so-called matrix of the mitochondria. And this just shows that if we increase patch clamp the mitochondrial inner membrane and increase the voltage, that the flux of protons increases when you get to about 180 millivolts. And if you knock out the adenine nucleotide translocator, you completely eliminate this. So in fact, all proton leak in all of our tissues, except for brown fat, is through the adenine nucleotide translocator. So what that means is that we have a system by which we have one force pumping protons and another force, which is rheostated, that releases the protons. And what that means is you pump up a capacitor and then you release it. And this, we are hypothesizing, creates an oscillating current. So to study that, my colleague Peter Burke designed this structure in which he puts carbon nanotubes and then, of course, some electrodes for which we can then measure voltage oscillation as the mitochondria flow down microchannels past these carbon nanotubes, and then we're measuring the voltage fluctuation. And of course, the first thing that happens is as the mitochondria cross the carbon nanotubes, you get a big deflection. But what's important is as it's sitting on the carbon nanotubes, you have this very rapid oscillation. You can see that here, or amplified here. And this oscillation, we don't know its frequency yet, where it's very relatively high as far as we can tell. So what that says is that when we have mitochondria here on these nanotubes, and we now add substrates, what you can see is that the mitochondria become polarized, and then they begin to oscillate by this electrostatic tie. So what we're really looking at then is oscillation of the mitochondria. So why is that of interest? Well, and this, by the way, is figures from a paper by Elserman and his colleagues, published in 2018. So if you now put cells into a magnetic field, say about 50 microtesla, which is the equivalent to the Earth's magnetic field, and now you add an electromagnetic oscillation, in this case they use 1.4 megahertz, either perpendicular to the magnetic field or parallel to the magnetic field, what you do is you increase the mitochondrial generation of reactive oxygen species. And this occurs from electrons that are donated directly to O2 rather than going through the electron transport chain to water. And so depending on the orientation of the oscillating field relative to the static magnetic field, you get different ROS production levels. And if you then look at mitochondrial respiration, and you look at a oscillating field that's perpendicular, that's parallel to the standing magnetic field, it doesn't affect mitochondrial oxygen consumption. It radically increases glycolysis. But if you put an oscillating field parallel to the standing magnetic field, then you change the oxygen consumption on the mitochondria without changing glycolysis. So you can see that just changing an oscillating field relative to a standing magnetic field, you have a radical effect on mitochondrial physiology. And why is that important? Well, the Earth's magnetic field is just about 50 microtesla. So we're sitting in a standing magnetic field, and if the mitochondria is generating an oscillating electromagnetic field, then we have exactly this situation inside our mitochondria. Now, these authors also were quite interested in the idea that the electron transport chain, which has a number of electron carriers, can pick up an electron, and then the electron can either be in one orientation or the other, and then a second electron can come in, and if it's, of course, anti-parallel, then it will go on to the electron transport. If it's parallel, if the spins are parallel, then in fact it inhibits the rate of that, and that increases the generation of reactive oxygen species. So their idea was that the oscillating magnetic field can affect this kind of electron flux. But there are many different carriers in the mitochondria for which this could be a possibility. So basically, our thinking, and again, this is hypotheses we've been working on for maybe 20 years, is that the crystalline encompass a very closed volume, less than 0.3 nanometers. And this is sealed in an intermembrane space. The electron transport chain pumps protons into the Christie lumen. The Christie and adjacent mitochondria are aligned, and we believe that's due to the positive charge electrostatic repulsion. The voltage-dependent ANT releases the protons at about 180 millivolts. Therefore, the electron transport chain and the ANT are creating an oscillating system which could generate an oscillating electromagnetic field. And due to the electrostatic interactions, adjacent Christie and therefore adjacent mitochondria and therefore adjacent organs might in fact be oscillating in phase. And then the second concept is that protons have a nuclear spin of 1/2, and the Earth's magnetic field provides a static magnetic field in which our mitochondria function. The lipid bilayers are so close that they, I believe, are limiting thermal oscillation within the Christie lumen. Therefore, the Earth's electromagnetic field could well align the spins of the protons. And that then could create a situation where oscillating electromagnetic field could alter the orientation of the spin, such as in a magnetic resonance imaging system. And then we have shown from the other people's work that oscillating this field in the static field will affect glycolysis, mitochondrial loss, and mitochondrial respiration. And then the last point is the electron transport chain moves electrons to a series of electron carriers that take one electron at a time, and that flavines being one example. And that could then create a situation of singlet, triplet interactions, which would regulate a series of reactions through Ross, both chemical and through signaling. And so that's basically the ideas that we're putting.
So my talk was about photobiomodulation, which is evolved from what used to be called low-level laser therapy, and now it involves putting visible, quite often red or near-infrared light onto the human body in order to treat all sorts of different diseases and conditions. And I was particularly interested in brain disorders. So the mechanisms of why this works, we've been researching those for some years. And last talk you heard a lot about mitochondria, and of course mitochondria contain many of the chromophores, the cytochromes that absorb red and near-infrared light. And it's not exactly clear which cytochromes are really heavily involved. This was cytochrome C oxidase, unit 4 of the respiratory chain, which has heme and copper centers. So its absorption spectrum goes right into the near-infrared up to 850 nanometers. And the idea was that the light could photodissociate inhibitory nitric oxide from the copper center. And once the nitric oxide is displaced, oxygen can come in and you get a big increase in respiration and ATP generation. Other molecular mechanisms have been proposed, included heat and light gated ion channels, transient receptor potential ion channels, which are known to be activated by blue and green light in terms of opsins. OPN form melanopsin. However, they also activated by heat, and near-infrared wavelengths can produce very localized heating in nanostructured water. So you get conformational changes, open the ion channels and get, obviously calcium is the main thing, and changing calcium in mitochondria is very important. And the third molecular possibility was this molecular rotor of ATP synthase you heard about in the last talk. And the idea is that if you can decrease the viscosity of the nanostructured water surrounding ATP synthase, it would turn faster. And if the molecular rotor turns faster, you would generate more ATP. So all three of these mechanisms, again, are centered on mitochondria. Really, I think everybody in the photobiomodulation field believes that mitochondria are at the very essence of why it works. But once you've activated mitochondria, you get a whole bunch of other things happening. So obviously you get more ATP From that, you can get more cyclic AMP, which of course can activate June phos and AP1, an important transcription factor. You can get a burst of ROS due to raising the mitochondrial membrane potential, which can activate protein kinase B and degrade I kappa B and allow NF kappa B to go to the nucleus. And then the nitric oxide produced can also do a lot of signaling. So you've got a lot of signaling pathways and especially transcription factors that are activated by light, which explains why a very brief exposure to light can have experimental effects that in mouse models can last for 1 month. So these are very long-lasting effects, which is different from what you think most exposure to light can do. So in the brain, these mechanisms have many, many effects. And as you might expect when you produce activation of mitochondria. So in addition to getting better oxygenation and more ATP, which is very necessary for neurons to function, you also get anti-apoptotic signals, you get antioxidants, you get anti-inflammatory pathways that are activated, you can activate neurogenesis, you can activate synaptogenesis, you can, if you have a brain injury, you can increase angiogenesis and decrease edema. And a whole lot of beneficial processes have been demonstrated in the brain. So at the moment, we don't really know which of them are most important for which brain disorders, but all of these have experimental evidence for. In addition to the brain, this photobiomodulation works all over the body. But actually any degenerative or injury disease, when things need to be activated, healed, or regenerated. You generally don't use it for cancer or infections because there's a worry you may be able to stimulate unhealthy processes, but in all sorts of degenerative or painful inflammation diseases, it has had remarkably good results. So the number of positive clinical studies is almost exponentially increasing. 20, 30 years ago, it was considered to be a bit of an alternative complementary procedure, and some people said it was junk science, but as the years are passing, the amount of really hard clinical evidence has increased, so it can't any longer be ignored. For the brain, there's been several devices devised to put red near-infrared light on the head, on the forehead. Hair is a bit of a shield, so you generally would like to shine the light on the area of the head without hair, but even so, some light, near-infrared light, will go through hair. Many of the pathways in the brain that are affected are common in both neurodegenerative and psychiatric disorders. So brain inflammation is bad, oxidative stress is bad, excitotoxicity is bad, mitochondrial dysfunction is bad, apoptosis is bad. And photobiomodulation can help all of these. It can increase BDNF, increase neurogenesis, increase synaptogenesis, prevent brain shrinkage, and treat all these degenerative and psychiatric disorders. So just a list here of all the different brain disorders that people have considered. A lot of sudden things, a stroke, a head injury, heart attack, difficult birth, these sudden things that deprive the brain of oxygen. You can help them by getting the near-infrared light on the head as soon as possible after the sudden disease. Psychiatric diseases are obviously more chronic, as are neurodegenerative diseases. So there, the light will likely have to be applied to the head, continuously, 2 or 3 times a week, every day, something like that, for about 15 minutes. Certainly for neurodegenerative diseases, that will probably be lifelong. For psychiatric diseases, you know, a course of treatment of maybe 3 months, something like that. There are some things in common between photobiomodulation and non-invasive brain stimulation in terms of the conditions that are treated, but there are differences, that the light doesn't really trigger action potentials the way that our TMS or TDCS actually does. But there are definitely things in common. And they may be able to combine photobiomodulation with these other non-invasive brain stimulations.
So I'm sorry you can't see me, but the most important thing is the topic, which is the unraveling of electromagnetic effects on or in living cells, one microtubule at a time. As you probably know, I'm really a big fan of microtubules, and here I'll just try to summarize our work and also say a few words about our future plans in this regard. This is a joint collaboration with Professor Aristide Dogariou from University of Central Florida, who is a photonic expert, and I'm a theoretician. I'm a biophysicist. So we work within the hypothesis that energy transduction in living cells is electrodynamically mediated, and a very important role is played by microtubules and microtubule constructs in this process. And these constructs are things like centrioles, cilia, flagella, microtubule bundles, and neurons, etc. So there is a large scope, including microtubule mitochondria complexes. So there is connection to the first summary today. And the processes where these things play a major role are cell division, intracellular transport, ion channel modulation, cell motility, and signal propagation. We are scratching the surface. Very little is known about all of this. We've just done a few years of work on electric signal propagation, electric conductivity, capacitance of microtubules and microtubule constructs, as well as electromagnetic responses in various frequency ranges and wavelengths. And all of this indicates that we should take very seriously a challenge to the central dogma of molecular biology that only short-range specific interactions play a role. We actually see quite a bit of long-range interactions. Most of it so far has been classical. We are currently working on quantum effects, but I'm not ready to disclose much of it yet. We'll come to it maybe a year from now or sooner. But as far as what we know now is quite rich and interesting. There are frequency-specific responses to both mechanical and electromagnetic waves of microtubules and microtubule constructs, and by this I mean tubulin, starting with tubulin to single microtubules and parallel microtubule networks as such in neurons, or radiating microtubule networks in dividing cells, and specifically arranged microtubule networks in centrioles. And so these frequency responses range from hundreds of kilohertz, which are now used therapeutically for cancer by Novocure, to megahertz ionic movement along microtubules, to conformational changes of microtubules in the terahertz region, which we just published. And we have data that will be probably very soon published on using Raman spectroscopy to demonstrate that these electromagnetic waves interact specifically with some structural features of tubulin and probably other proteins as well. So we are getting a lot of information about how this is happening, and all the way to the near visible and visible ranges of electromagnetic waves. So what we can conclude about microtubules and electromagnetic fields is that, first of all, and I will not repeat the specific findings from my talk last month, but there is a very rich behavior of individual microtubules, which to us, surprisingly, and maybe to others not surprisingly, is very strongly dependent on the medium. The ionic concentration in particular, as well as pH changes, affect conductiviity, capacitance of microtubules, and also responses to electromagnetic fields. Some of the responses are catastrophic, that is depolymerization of microtubules under the influence of pulsed terahertz fields, or some are actually more favorable changes in the polymerization rates, and in some cases actually even improvement of polymerization. So we still need to find a very quantitative description of these processes. Most importantly perhaps is that microtubules can be either conductive vis-a-vis ionic currents within the cell, or capacitive vis-a-vis storage of ions, or becoming basically charge storage devices. And this transition is concentration dependent, ionic concentration dependent. So we're moving towards predictive quantitative models of electromagnetic energy transduction, starting from microtubules. And so this is a kind of busy table with all our constructs that we want to develop, one microtubule and other things at a time. Single microtubules, bundles of microtubules, then microtubules with motor proteins. We don't have evidence yet, but we have a strong suspicion that motor traffic is very much affected by both electric and electromagnetic fields. And then definitely things like centrioles and higher organizational structures with microtubules may be very sensitive to electromagnetic field effects. And so this is the sort of scheme of things where we are going with it, in vitro constructs. And I want to link all of this to some of the effects which were mentioned by others, in particular mitochondria, the activity of mitochondria, which actually move on microtubules via motor proteins, and very much membrane transmembrane potentials have an effect on the ionic concentrations and pH, and lead to pathologies and so on. So there's a huge amount of, a huge scope of connecting this to Mike Levin's work and others. And then we have the currently being developed experimentation work on tryptophan networks. So we are moving towards quantum effects, absorptions of photons and re-emission, and quantification of this depending on the geometry where we have actual evidence for that happening. And then towards, as I said, more complicated structures like centrosomes, which have been touted as the eye of the cell and maybe the brain of the cell as well. And finally, with Dr. Dogariu's help, we are developing diagnostics for a single cell using power spectrum analysis for the full cell dynamics, which could be very useful in testing, for example, new drug entities as they, in a very precise quantitative way, linking the concentration of the drug to the cell dynamics, and that can tell us about the precision medicine in terms of concentration and scheduling and dosing and things like that. So I will stop here, and I thank you for your attention, and we'll stop sharing.
Jack, thank you very much indeed. Extremely succinct, and thank you for showing your slides there. Margaret, can we go over to you, please?
Okay, hello. So I'm going to talk about infrared and electromagnetic field as possible therapeutic tools for treating the SARS-CoV-19 epidemic. And what I mean by that is not as an antiviral agent, but as an agent to deal with the inflammatory response pathway, which is the leading cause of mortality in this disease. And we have a cell culture model system which perfectly replicates the inflammatory response triggered by this virus. And the first question was, could electromagnetic fields be used for therapeutic purposes? And there are actually many of these kinds of devices already on the market. We took one single modified device and looked at a very simple pulsed field signal of 10 Hertz, 2 millitesla peak intensity. And we did a 10-minute exposure of these cell cultures twice a day for over just 48 hours. And we looked at the inflammatory markers in the control stimulated cells and in the treated cells. And there was a very nice and significant decrease in the inflammatory response. So how could this be? We also noticed that these pulsed field devices are inducing reactive oxygen, bursts of reactive oxygen in cell cultures. So these are cell cultures treated with a fluorescent dye, which turns bright green as a cause of production of reactive oxygen when exposed to these pulsed field devices. And so our hypothesis, our current model for how these magnetic field effects work is that they trigger a physiological burst, a transient burst in reactive oxygen, which induces the anti-inflammatory response pathway. And so the trick is to get the right dose in the cell, not too much, not too little. And I think we heard a nice talk by Doug Wallace on how these weak magnetic fields could induce ROS. And the physiology of inflammation and the response to reactive oxygen is well studied and documented. So that was one part of what we looked at. The second was photobiomodulation. Could that also be effective against COVID-induced inflammation? And where did this come from, this idea? After the nice talk by Mike Hamlin, you now know these kinds of treatments have anti-inflammatory effects. They also generate reactive oxygen, likely through stimulation of mitochondria. So again, we used our model cell culture system. We induced the inflammatory response, and we treated twice a day for 10 minutes with this infrared light, and over a period of 48 hours. And again, here is an ELISA of the cytokine secretion. So this is the worst, the most dangerous inflammatory cytokine. And these are inflamed cells, and these are treated with infrared for 2 days, and the decrease in secretion was by 80%. So this was very dramatic. We also took a look at the actual alveolar epithelial cells, human lung cells, and resident macrophage cells. So these are the actual cell types that are affected by cytokine storms, by this hyperinflammatory response. And in both cases, this gene expression, the cytokine levels were decreased. I'm summarizing a lot of data which we did along similar lines. Finally, I want to end with some preclinical data gleaned from the literature with 12 patients that various doctors, 12 COVID-19 patients, pre-ventilator, late-stage patients, treated by several doctors and using lasers once a day for a few days, so a version of photobiomodulation. And the result was that all of these 12 patients got better. When they did a matched group, the treatment group, which was treated, 0 patients ended up in the ICU and 0 patients died, whereas a matched control group at the same stage without photobiomodulation treatment, 6 patients ended up on ventilators, of which 4 died. So given these encouraging, very encouraging results, both in the lab and some preclinical data, I conclude with the suggestion that if proper clinical trials, credible and large-scale trials can be conducted, these methods, either photobiomodulation or magnetic field therapy, could actually be very useful. And in the treatment of COVID, they are affordable, they are safe, they are effective against any variant of the virus, and they can be implemented immediately. I mean, if a patient can go home 2 days after he ends up in the hospital, after this kind of course of treatment, I think then the epidemic is no longer a great concern. So that was a summary, a resume of my talk. And I think that I'm very happy the other speakers completed many of the fundamental concepts which I applied in this trial here. Thank you.
Margaret, thank you so much again for showing us this very intriguing finding and quite fascinating as to where the next steps might go. Can we now turn to Christoph? I think, Christoph, you have for us a single slide.
Yes, that's right. I just have to find it now. Here, can you see that?
We've got it. Thank you.
Okay, excellent. So my talk was the most recent, so I thought I would keep it short. So I had, I guess, two main themes. Like one was biophotons, and then the other one was radical pairs and spins and so on. And I'm really interested in potential connections between those two. So on the photons, so we know, of course, that there are biophotons, and let me mention right away that there is evidence that they probably come primarily from the mitochondria. So there is right away a connection to Doug's interests and also to Margaret's interests, because it looks like reactive oxygen species are likely involved in the biophoton generation. So we are interested in the possibility that these biophotons might serve as signals, either classical signals or maybe quantum signals in the brain. And our, I guess, main contribution so far was the suggestion that axons in the brain might serve as waveguides, serve as photonic waveguides for these biophotons. We did a lot of theoretical modeling on that, and in my talk recently, I also showed you some experimental evidence now that is indeed the case. So that could sort of be the optical fibers in some kind of network that we might have in our heads. Then I said, okay, if we are thinking in terms of networks, we also need nodes of some kind, and certainly for a quantum person, like spins would be very attractive nodes. So that motivated me and my group to start looking a lot at spin effects and magnetic field effects in the brain. And the first paper we wrote on this was this paper on anesthesia, where we showed, again with theoretical modeling, that's what we do, that a radical pair-based model can explain a number of experimental findings on anesthesia that people have made, and in particular isotope effects. So people have seen isotope effects for anesthesia with xenon, and we can sort of quantitatively explain how that might happen if radical pairs were involved. And then what we realized then over the following couple of years is that doesn't seem to be an isolated phenomenon. Like there is a lot of some isotope effects, but most of all, a lot of magnetic field effects, like in the slow field range around the Earth's magnetic field, that people have seen in biology. It's really an almost not unending list. Like there's dozens of these papers in different systems, and we have only had time really to scratch the surface, but we wrote a few papers, and we always conclude that radical pair models could be the explanation. So for sure we need more evidence and more experiments to show that indeed they are the explanation, but it looks like they could be. Very often reactive oxygen species are involved. I think in all our models in the papers that I listed here, our ROS are partners in the radical pair. So there is a strong connection here to Margaret's interests, and also to Doug's, I think, because so the ROS, we know that the two main sources of ROS, and at least that's my understanding, are the mitochondria on the one hand, and then NADPH oxidase, NOx, on the other hand. So mitochondria certainly seem to be important. But it does go beyond cryptochrome. So radical pairs have been studied a lot in the context of avian magnetosensing and cryptochrome, and Margaret is one of the experts for that, but it seems to us that really these phenomena go beyond that. So that like Doug was suggesting on one of his slides, that other flavins maybe and certainly ROS, seem to be involved, other mechanisms play a role. So it would really be good to get a more global view. I think that's one of my aspirations now, to get a better understanding of all the ways in which this radical pair phenomena, which is really a quantum phenomenon, it plays a role in biology. But anyway, my concrete examples were lithium treatment of mania, where we think we can explain an isotope effect and we predict magnetic field effects. Then we had results on the circadian clock, again, magnetic field effects. Microtubules, JACs, big interest. We think that there's magnetic field effects that people have seen, again, in this low field range. And we think we can explain those if radical pairs are involved. And then an impressive paper that showed how shielding the magnetic field, just shielding it, had a strong effect on neurogenesis and cognition in mice. And we think we can understand all of that with these radical pairs and ROS. And then so what I'm really now interested in too is, if one can connect. So you can see that I mentioned ROS here in the context of the biophotons. ROS seem to be important here in the context of the radical pairs. It looks to me like, so I'm a network, quantum network person. So I'm looking for an interface, right, between the photons and the spins. And it seems to me like ROS could be such an interface. And if you talk about localization in the cell, certainly mitochondria could be a candidate system, because we know that there's evidence, that that's where the biophotons come from, and there's evidence that they are important in this radical pair context. So if you give me that, so if there's a node somehow, or if there are nodes where photons and spins can talk to each other, then I could imagine sort of a quantum network that could create large-scale entanglement in the brain, and that's something that I'm very interested in the context of trying to understand consciousness. That is the most speculative part, of course, of the whole story. So I'll leave it at that. Thank you.